Hello everyone, my name is Jayesh. So today we will learn how to make a 3D model of DNA at home. Let us first understand what DNA is. DNA or deoxyribonucleic acid is the hereditary material in humans and almost all other organisms. Nearly every cell in a person's body has the same DNA. The information in DNA is stored as a code made up of 4 chemical bases: adenine, guanine, cytosine, and thymine. DNA bases pair up with each other, adenine with thymine and cytosine with guanine, to form units called base pairs. Each base pair is also attached to a sugar molecule and a phosphate molecule. DNA is a double helix formed by base pairs attached to a sugar phosphate backbone. To make 3D model of DNA, we need some colored paper, earbuds, glue, and some sketch pens. First, we need to cut the colored paper into 2 thin slits, which will serve as a sugar phosphate backbone. After making 2 slits of paper, we need to connect the earbuds into those papers with the help of glue, as shown in the video. These earbuds will represent base pairs in a DNA. After connecting those earbuds, we will color an earbud in 2 different colors into the half, which will represent adenine, thymine, guanine, and cytosine on base pairs. After attaching base to our structure, we will twist the DNA to form a spiral double helix structure. Now, our 3D model of DNA is ready. Hello, my name is Jayesh Ghodvinde and I will be telling the importance of herbarium. So let's go. So what is herbarium? Herbarium is a botanical research. It is collection of preserved plant specimens inked and dried on an herbarium paper along with all necessary information required by researchers, students and general public. An average herbarium is based on levels of classification which are in turn based on morphological, anatomical and habitual similarities. So let us see the history of herbarium. The idea of herbaria was first developed in Italy in the 16th century, where dried herbs were gathered on a paper at that period. Luca Guinea invented the herbarium. The herbarium specimens might be carried from one location to another. This made it possible for doctors to use these samples as reference for material when creating herbal medicines. So what are the functions of herbarium? The preserved plant specimens from herbaria are used in majority of taxonomical research projects, such as primary source of getting knowledge regarding the variety of plants and flora. A list of plant species that include their medical and other ethnobotanical value can be created after consulting an herbarium. The third point is all of these species in a genus or all of the genera in a family might be represented in the herbarium. And the last is the herbarium material can be utilized for studying the chemistry, anatomy and palynological desired plants. So what are the uses of herbarium? The basic functions and research. Herbarium can be used for basic functions and research as well, where we can find out a plant's identity, confirm it or establish that it is unknown to science. It could also be used to verify the nomenclature for plants' Latin names. And it also includes providing data for DNA analysis in the areas of systematics, evolution and genetics. The second use is in education and training. It provides instructional materials in forestry, agriculture, dendrology, field botany, taxonomy, and ethnobotany. It also introduces systematic research. By making specimens available for viewing by students, researchers, and general public, increases appreciation of plant diversity. So, what are the steps involved in this process of herbarium? The first is collection. Botanists and plant enthusiasts collect plant specimens from the wild or cultivated gardens. These specimens often include leaves, flowers, stems, and sometimes fruits or seeds. It's important to note the location, date, and the other relevant information when collecting specimens. The second is pressing and drying. Once collected, the plant specimens are carefully arranged on sheets of absorbent paper and pressed to remove the moisture. This process preserves the plant's shape and structure. The sheets are then dried to ensure long-term preservation. The third step is mounting. After drying, the specimens are mounted on sheets of archival quality paper, and the labels with essential information about the plant, such as its scientific name, common name, collection date, location, habitat, and collector's names, are attached to the sheet. And the last process is of storage. The mounted specimens are stored in cabinets or drawers in a herbarium. The storage conditions are controlled to prevent deterioration, such as exposure to light, humidity, pest, and temperature and fluctuations as well. The herbarium collected by me is the damask rose. You can see the herbarium mounted on the sheet in the photos below. The date of collection is 15th of June. The plant is damask rose. Its scientific name is Rosa damascena, and all other species details could be seen below. Thank you.